and welcome to our next talk here at Pycon. We're going to hear from Jack Reichelt about Walking the Tree of Life, Adventures in Phylogenetics. Thank you, Jack.
G'day everyone, and thanks for coming to my talk. So, I am Jack Reichelt, wearer of too many hats. I'm the CTO and co-founder of a startup called Kumo Study. We make a study management tool for students with a focus on students with ADHD. We have sort of a prototype version. We're launching the real version 2 come March-ish next year. I am big into computer science education. This is actually my first Pycon talk that is not part of the education track. And most relevant to this talk, I am the holder of a wide variety of ADHD-given interests. So now that you know a little bit about me, we're going to go back to that title slide because this talk has a subtitle, which was mentioned, The Adventures in Phylogenetics. It also has a subtitle, which was more recently added and not on the program, How It All Went Wrong. So this project didn't end up where I expected it to, but along the way I found some interesting stuff, I learned a thing or two, and I hope there's something you'll find of value here. So, I promise that we will get to the computer science, but first we're going to take a detour through some other sciences in order to understand what I was trying to do and why. So let's start by clarifying what is phylogenetics. So, phylogenetics is the study of life and evolution using genetics. Specifically, it's not just saying these two organisms seem alike, so we'll group them. It's using molecular analysis of DNA and empirically cataloging and categorizing organisms. Remember back in school in your science class when you learned about taxonomy, the whole domain, kingdom, phylum, yada, yada, yada? That's what I'm going to call vibes-based classification. As an example, giant pandas were named as such because of their obvious similarities to red pandas, which were just called pandas at the time. In actuality, giant pandas are a type of bear, and red pandas are more closely related to skunks and raccoons. Now, we later corrected this and realized that giant pandas are really weird bears, but the whole process was still pretty non-scientific. Also, it's a bit weird to assume that every organism just has 8 layers, right? Like, if each layer is a divergence from other organisms, that doesn't make a lot of sense. Has every organism only experienced 7 divergences? And what about the earliest organisms? How had they experienced 7 divergences? Or did they not have a bunch of these? Was it just species blank for all the rest? And if it's that one, then how long until we have to add a new tier, and where does it fit in? Are you going to add something below species? I mean, I guess that's kind of subspecies, but that's not even on this list, so taxonomy's a bit weird. Maybe it's not very good. So we're going to use clades. What is a clade? A clade is basically just a group. Nothing more to it. Each clade can have other species within it, or if you're generalizing outside of organisms, then just leaf nodes, or it can have other clades within it. And it's clades all the way up. Just every successive tier up is always called a clade. Humans are the species sapiens, which is inside the clade Homo, which is inside the clade Homininae, inside Homininae, and so on, all the way up to the clade Biota, which contains all organisms for the last 4.2 billion years or so. There were actually some really early ones that weren't in biota. They all died out. There's none of them alive. Everything came from one branch now. So, these clades, in contrast to the taxonomy groupings, are science-based. They're formed using genetic analysis and the knowledge of the mutation rate of DNA. So you can analyze the genomes of different organisms and tally up the differences, and then count how many there are, and that tells you how long ago they must have diverged. You can use these divergences in order to plot them all out and categorize them all. So what is the point of all of this? Why ultimately would we bother doing this? Well, for starters, it's more correct. Correctness is good and important in science. It is worth it just for the better understanding that comes with it. But it can also help us in more concrete ways too. It can give us a much more thorough understanding of how organisms are related. This can be useful for all sorts of things, from veterinary sciences, to rearing and taming, to medical research, to not naming animals pandas that aren't pandas. It can also help us understand the whole evolutionary history of an organism. If we know when it diverged from other organisms, we can infer when certain traits must have evolved, and that can help us place extinct animals or any other animals that we don't have the DNA for. So what was I trying to do? I wanted to make a program that would allow me to explore a phylogenetic tree of life, also called a cladogram. I thought that, assuming a reasonable data source somewhere, I should be able to make an interface that let you click around and see what the different relationships are, see what organisms are in a clade, find their closest relatives, that kind of thing. Ultimately though, my main goal, my true desire, was to be able to ask how closely related are these two things, and what animals, or what species, I'm mostly interested in animals, but it works for anything, what species are in that clade specifically? Remembering that a clade can both have clades and leaf nodes within it. So, seeing the smallest clade that contained them both, and what clades are in, what species were in there. From a computer science perspective, this doesn't sound too hard. Ultimately, this is just a tree like you've probably worked with before. Finding a subtree is not too hard, nor is finding a shared parent node, assuming a reasonable data source. So, if this is so simple, surely it exists already, right? Well, you'd think that, wouldn't you? But I hunted around and found a variety of tools that were not quite what I wanted. I found ones that had many of these features, but you needed to upload the data in a specific format that you already had. I found ones that didn't have lots of the features I wanted, and I found one that did almost everything I wanted. It had a great interface, it had all of life, it had a good search function. The only thing it didn't have was a way to give me a way to ask how closely related are these two things and get all that information. We'll come back to that one later. So having established that nothing did quite what I wanted, I thought to myself, hey, this sounds actually useful to real scientists though. Surely somebody needs to at least occasionally say how closely related are these two things. Maybe I should ask a biologist. So I reached out to the only practicing biologist I know, my uncle. Here's a brief introduction to my uncle, Dr. Russell Reichelt. He has had many notable achievements in his lifetime, and here's just a few of them. He's a director of the Great Barrier Reef Foundation. He is a board member of the Australian Climate Change Authority. He is an officer of the Order of Australia, an all-around top-notch bloke, and most importantly on his list of achievements, he's my uncle. So, I wrote him an email and laid out the idea that I had, the investigation that I had done, a few questions, and asked if he thought it was worthwhile. And he basically said, I don't do any of that kind of stuff anymore. I mostly deal with policies and politics. You need to talk to Dr. Pat Hutchings. Dr. Pat Hutchings is a senior fellow at the Australian Museum and has done extensive research on the ecology and phylogeny of Australian polychaetes, which are cool little undersea worms. So I had a great phone call with her, and she gave me some good advice and shared with me the most unforgettable sentence anybody has ever said to me. She said, "Tim Flannery would be a good person to talk to about this, but he's a bit busy, so he might not have the time." Now, for those of you who don't follow this sort of thing or are too young, Tim Flannery was Australian of the Year in 2007. While he would no doubt be an excellent person to talk to about this, he is also, unfortunately, a bit busy. It was then I realized I had made a tactical blunder. In my head, what I had done was present my idea to the closest biologist at hand and ask, hey, is this a good idea? Can you answer some questions for me? In reality, what I had done was present my half-baked beginnings of an idea to an extremely distinguished scientist who had generously introduced me to another extremely distinguished scientist who had, in all sincerity and with the utmost of kindness, tried to find the best person to answer my questions, and so she recommended the Australian of the Year from 2007. I did not reach out to Tim Flannery. Instead, I spoke to Dr. Jacqueline Nguyen, who Dr. Pat Hutchings also recommended. She is a postdoctoral researcher at the Australian Museum, and she is focused on the evolution of birds. And she was recently involved in a project reworking the whole phylogeny of all birds, a very relevant project to exactly this kind of investigation. Jacqueline was a huge help. She answered my questions, she pointed me at some potential data sources, and generally confirmed that being able to see how two species were related would be a good tool. And so, armed with her advice and some confidence that this was a good project, I started diving into the meat of the project and pretty quickly hit some snags. The first data source I looked at was ITIS, the Integrated Taxonomic Information System, and it had many positive features that looked helpful. You could search by common name, which seemed ideal for enabling casual use. It had really clear documentation, so it was easy to access their API, and it even had a whole database dump available for preloading all the data. However, it did have a couple of problems. The first problem was that searching by common name was tricky. There are so many common names. Suppose you search for hedgehog. You probably meant this, right? Or did you maybe mean this? Or this? Or one of the 154 other options that it presents when you ask for hedgehog? There was also no way to filter these results, nor are they sorted by anything other than ID. There was nothing to do except give the user every single option and let them pick. And that's bad enough for hedgehog. Imagine trying to pick from the 355 different carp results, or the 472 sharks, or the over 1,000 worms. This relates to the second problem I had. The documentation was very clear, but the API was sadly not. Suppose you had found the species you wanted, and you had its ID. So then you query the API to get the parent clade for it. What returns is the ID for that clade. So then you have to do a new API call to get the actual data of that clade. Okay, that's not too bad. Let's try the reverse. You have a clade, and you want to find all of its child clades and species. So you query to get the children, only it returns the whole subtree underneath that clade, and also only the IDs. So now you have to make an API call for every ID there, and check if it is a direct child. And then, for those ones, make another API call and get all of their details. To compare just two quite related species, like say a seal and a ferret, you have to make over 100 API calls, and it wasn't particularly fast. Each of them took about a second. And that's after you've picked from the 12 ferrets and 144 seals. Or things that return when you search those things. But that's no problem, right? There's a whole database dump, so you don't actually have to make these API calls, right? You can just load and read the database, right? Well, that brings me to the third problem, which is one less about the data source and more about me. I found that 4 days ago. So let's try the second data source. This is called the Open Tree of Life, OTOL. This data source seemed great. It was super fast, and it could do batch operations. So you say, get me all of this stuff for all of these organisms. And it could even be given 2 or more species and just give you their most recent common clade, or a representation of the subtree that contained all of them. That sounds fantastic, right? Well, it actually was, mostly. It couldn't do any common names at all. But I figured maybe this was fine. The common names were tricky with ITIS, so maybe that was something I didn't need, at least for the first version. So, with the data source decided upon, it was time to get to rendering and representing that data. So let's have a look at what I'm trying to draw and see what I'm trying to do. This is a cladogram of all of the different living bears. You can see here we've got a pretty classic tree structure that you've probably all seen before. We've got a clade at the root node there, that's Ursidae, and then each branch shows us different clades until we get down to the different species. Not every clade necessarily has a name. Often, if you dig deep enough, people have given these names, but they'll all be sort of extremely similar to Ursidae and Ursinae and on and on and on. You tend to stick to a few big ones. This cladogram is from a YouTube channel called Clint's Reptiles. Clint is a biologist who does a lot of videos about phylogeny, not just for reptiles, and it's how I got interested in all of this to begin with. So that was kind of what I was imagining making, at least visually. I'd like to have some interactivity, but this is what it might look like. Let's take a quick look at a different style of representation, though. This isn't a cladogram, but it's a different style of representing the same things. And it's a big fractal display where you can zoom in on each branch and keep zooming in and see what's there at every level. So we'll zoom in to the bears and compare that. So this is the same data as the last one, all the same species, represented with on this platform. So this representation is from one zoom. This is the tool that I mentioned earlier that does almost everything I want. And I think it's a great tool. It's really great for just looking around, seeing what life there is. These are two very different styles, and while both are good, sticking with the classic cladogram seemed like a simpler place to start. So I started by using a library called BioPython, and they have a module called Phylo, which is explicitly for phylogenetics. Okay, let's generate that same bear tree. Oh, I guess it draws it in ASCII. That's super cool, actually. I really like that, but it's not quite what I want. So let's try it again. All right, it's not ASCII anymore, but there are two problems with this. Firstly, this is way more bears, right? There were 8 before, and now there's what, like 40? So what gives? Well, partly, this is because Open Tree of Life includes subspecies. You can see wherever it's got 3 names, that's a subspecies, like Ursus arctos horribilis up here. That's the grizzly bear, which is a subspecies of brown bear. It's also got extinct species, like Ursus rossicus, the Pleistocene cave bear up there. Now, including subspecies and extinct species is kind of cool. I'm not actually opposed to it, though maybe it should be a toggle, because it does fill things up quite a bit. There's a bigger problem, though. This is just a PNG. So BioPython uses matplotlib to render its phylogenetic trees, and that just makes images, which is great for putting in a research paper or putting on a website or something. But at this point in the project, we're getting to crunch time. It's about 3 weeks out from Pycon, and I realized that if I'm going to make something that lets you actually explore these trees, it's going to need something a little bit more substantial, like an actual GUI. So cue a bit of panic. Okay, a lot of panic. I'm now certain that I can't make anything even remotely like what I wanted to do before Pycon. And then I made another blunder. I went and I looked at one zoom again. See, when I had done all of the research and talking to the scientists and everything, that was actually way back at the start of the year. Then this project sat on a shelf for a while I moved house, got married, started a CTO, and generally had an extremely busy year. Remember when I showed you this slide before? Did you notice this button up here? Now, I don't know when that button was added, but it either wasn't there when I first looked, or I didn't look very goodly. Probably that one. Anyway, look what happens when you click that button. Ooh, 2 entry fields. So what happens if I enter hedgehog and seal? Wow. Colored lines showing each lineage, and it shows where they meet. So let's zoom in on that, and oh, Laurasia, the clade that specifically contains carnivorans and also hedgehogs. Yeah, that sounds about right to me. So, where does that leave me? What happens to my project now, and what will I do with it? thanks for coming to my Pycon talk. No, I'll be serious. Truthfully, I don't know. In some senses, it might be worth continuing. Just because OneZoom is really good doesn't mean I couldn't also make something good. Or I could try and contribute to OneZoom, which is open source. Or maybe I'll just focus on that startup that I'm CTO of. But I can't just leave without showing the answers to the questions I started with in the abstract. So, which animals are more closely related to cows than pigs? Everything along this red line, which includes deer, giraffes, and whales. Are birds reptiles? They sure are. If they're not, then neither is anything along the blue line, but not the red line. So that includes turtles, crocodiles, and dinosaurs. If you're willing to say none of those are reptiles, you can say the birds aren't reptiles. But if any of those are reptiles, birds have to be as well. Am I a fish? Yeah, probably. So there isn't a specific fish clade. So what we can do is what's called phylogenetic bracketing, where you pick two different organisms that share the trait that you're looking for, and look at where they meet. So in this case, if we think of a reef fish, like a clownfish, Nemo, and then any cartilaginous fish, like a shark or a ray or a sawfish, if both of those two things are fish, then everything where those two lines meet at the jawed vertebrates must be fish as well. I have a jaw and a backbone. I'm a jawed vertebrate, and if you look way closely over there, just in the top left, it says mammals. Yep, we're fish. And lastly, what is the last common ancestor of a bear and a weasel? Well, actually it shows us a clade, but it doesn't show us any species within that clade. So I guess OneZoom doesn't do everything that I want it to do. So maybe there's still some room for my project after all. Okay, thanks for coming to my talk for real this time. I hope you learned how the best laid plans of fish and men can go wrong, but maybe still be worth it. I hope you learned something neat and interesting about modern biology, and I hope that you're inspired to just try stuff out and then try talking to random experts and seeing how you go. You never know, and maybe I'll send a link of the recording of this to Tim Flannery. Thank you very much.
Thank you very much, Jack, for that delightful talk. Do we have any questions? We have a question.
Can you say it in a big word? Big voice.
This is Renee and me, and Jack and a bear, and a
Alex,
kid.
did you have a
Was there-
question for Jack rather than a comment? Can you phrase your question as a question?
Yes?
Why do pterodactyls fly?
So the question was, why do pterodactyls fly? A big reason why pterodactyls fly is because the sea levels were a lot higher at the time when pterodactyls started to evolve. And so instead of lots of big land like we have now, there was lots of little islands, and they needed to get between the islands. And a really good way to get between islands is flying. And so a lot of the pterodactyls, especially the really big ones, they started flying because of that. So to get between islands.
To get to the other side.
So you're a CTO. That's pretty fancy. A lot of things went wrong in this talk. Well, not in the talk. The talk was great, but the project had some hiccups. How did you, as an established developer, come through those hurdles, especially like from the perspective of someone who might like be a new developer? How can they approach difficulties like that and keep on going?
Yeah, good question. So I think it really depends on what you're trying to do. Ultimately, I was making this project because I was just messing around. I think that all this phylogeny and organisms and biology in general is super interesting, and so I just like messing around with it. So I didn't necessarily need to keep going, and when I hit snags, I was more than capable of just dropping the project, except for the fact that I had long since submitted to Pycon and got told that I was going to talk, so I needed to come up with something. But if I was doing this for a uni assignment or in my work, I'd probably think about like taking a step back and asking myself, what are the problems I'm actually trying to solve? What am I getting stuck on? Is this something that I can put to the side and come back when I've learned more about the problem space? Is this something that I can actually just circumvent and solve other easier problems? Is there somebody on my team or in my general network that might have an answer that I can talk to and get some advice from? Talking to people is underrated and really good. Would recommend.
Is that Tim Flannery?
Yeah, would chat to Tim Flannery. Absolutely, I would chat to Tim Flannery if he was not so busy.
Thanks, Jack. Sorry. Thanks, Jack, for the great talk. It inspires me to give a talk at Pycon too. Would you consider repurposing your software to find clades between languages?
So, I do think that that's quite interesting. And I was, when I watched the languages, in fact, both of the previous talks in this room, I was thinking about the similarities and the clustering and the relationships between the languages and things. I think that could be very interesting. I wonder how much of it would be sort of new and novel, because relatively speaking, there's much fewer languages than organisms. And so maybe that clustering is like, that categorizing already exists. But I'm also seeing a head shaking from somebody who knows things. So maybe it doesn't exist and it could be useful and interesting. So yeah, maybe that's an avenue.
Hi. Was there anything else in your chat to the scientists that they came up with that they wanted to do that was not part of your plans at the time?
So a couple of the big things were that, as a slight, maybe not quite answer to your question, one thing that I've not seen in many phylogenetic representations, but is a big problem, is horizontal gene sharing. This is not a monodirectional tree. Species can and do crossbreed in the wild, even surprisingly distant ones. When I first talked to my uncle about it, he said that one of the big problems is like, in the past sort of 4 or 5 years or something, we found an instance of two unrelated species of coral become sort of hybridizing and making a new species of coral, and how do you fit that into there? And then you say, well, okay, what's the last common ancestor? And you're like, that gets a bit tricky. So I think that kind of thing is something that isn't shown, for example, in one Zoom, and might be something worth exploring or adding if I continue on with this and make my own representation. That kind of hybridization events and things.
Well, if you would join with me, please, in thanking Jack one more time for that talk.
Thank you very much. Thank you.